Okay, thank you everyone again for joining this webinar regarding single molecule long read 16S and 18S sequencing of microbiomes presented by Loop Genomics. I am Tuval Beni-Heskel. I'm the founder and chief scientist here at Loop Genomics, and I will be walking you through a presentation today that will hopefully give you a good sense of the technology behind loop genomic sequencing, as well as how we're applying them to single molecule long read sequencing of the 16S and 18S genes, as well as other microbiome applications like shotgun metagenomics and whole genome sequencing. Just a few procedural matters before we start. I'd like to mention that I would like to give you, the listeners, the opportunity to ask questions. Please write your questions in the chat section of the WebEx, and we will try to address each and every one of your questions towards the end of the talk. In the meantime, during the call, since there are quite a few people on the WebEx, please reserve your questions to the end, either through the chat or by asking them through your mic. And in the meantime, please mute your mics so everyone can enjoy the best quality audio during the talk. Now, I'd like to say a few introductory words about Loop Genomics before we head into the talk. So Loop Genomics is a single molecule long read DNA sequencing company. We're what's called synthetic long read sequencing. So in contrast to technologies from companies like PacBio or Oxford Nanopore, we have not developed our own single molecule long read sequencer. So we do not develop any hardware. Instead, what we do is we have developed technology to transform existing short read sequencers from Illumina into single molecule long read sequencers through the use of sample prep chemistry and software. Okay, so I think we can start this talk after these words of introduction. And please mute yourself, mute your microphone until the end of the call, so that the rest of the participants can enjoy clear and quiet audio. And then towards the end of the talk, you can unmute your microphone and ask questions either through audio or through the chat section of the WebEx. Thank you. Okay, so we here at Loop Genomics are super excited to be part of this new microbiome revolution. We think that this is a very unique moment in time to be studying the microbiome. We think that the microbiome is a very unique organ in the sense that it is a genetically encoded organ as any other organ in the human body, but in contrast to any other organ in the human body, the genome that encodes that organ, the microbiome, is not a static genome. So as opposed to our somatic genome, which is more or less static throughout our lifetime, our microbiome genome, the collection of all the genomes in our microbiome, are dynamic throughout our lifetime. So this is a genome that we possess throughout our lifetime, and it is dynamic. It changes from day to day. And that has never been the case for any other human genomic application. And this poses new and exciting challenges to the world of DNA sequencing, and we're excited to be in this space. One of the other reasons that this is so exciting is because, in contrast to other organs that have a static genome, for example, are the brain or the liver or the kidney, the microbiome is a hugely important organ in the human body, and its genome can be replaced. So in contrast to the genetic elements that determine the makeup of our brain or our liver, the genetic makeup of our microbiome is actually not only dynamic, but to a certain extent replaceable. So these two combinations of this organ having a dynamic genome and a genome that can be replaced means that the microbiome domain will not only offer us great insight into genetic diagnostics based on reading our microbiome, but it also offers many opportunities in the future in terms of therapy, because we already have a very good sense of the health effects that the microbiome can have on humans and on animals and on plants. So a natural consequence of that is that if we're able to replace or modulate that dynamic genome in a predetermined manner, we will also be able to affect human health. So those are kind of the major reasons that we're super excited to be operating in this space. And it's important to recognize that the reason that this space is now moving ahead so quickly is because there are technological advancements that are enabling this. And primarily these advancements are in the realms of DNA sequencing and bioinformatics. Please mute your microphones. So we think that in the past decade, there's been a perfect storm related to DNA sequencing. Since about 2007, 2008, there's been an exponential decrease in the cost of DNA sequencing per base pair due to the introduction of next-generation sequencing technologies. And in the same time, there's been an exponential increase in our ability to compute, as shown by Moore's law here. So these two are the two essential components that are needed in order to advance microbiome research. On the one hand, sequencing getting exponentially cheaper over time, and on the other hand, compute time or compute cost getting exponentially cheaper over time. These two elements are the two fundamental elements that synergize and to basically enable this big bang in microbiome research. So we're lucky, I guess we're all lucky to be working in this domain in this period of time where there are the technological advancements that are enabling us to both read this new and exciting genome and also do something about it by replacing and modulating it. Ironically, the same technology that has revolutionized next-generation sequencing, namely short-read sequencing from Illumina, is also the same technology that is now, to a certain extent, I'd say, limiting our view of the microbiome. So we know a lot of what we know about genetics today and about the microbiome today through the glasses of Illumina. So we understand whatever can be understood by reading DNA 150 base pairs at a time. But we all know very well that biology is not encoded in 150 base pair fragments. Biology is encoded in fragments that are thousands and tens of thousands, and sometimes even millions of base pairs long. So with current short-read sequencing technology from Illumina, we're largely blind to that type of information. And that type of information is extremely important, specifically in the microbiome domain. And the reason for that is because microbes are often very, very similar to each other. And just by reading 150 base pair fragments, it's very hard to tell which microorganism you're looking at, whether it's a 16S molecule, an 18S molecule, a fragment from a shotgun metagenomics experiment, or even an RNA fragment from RNA transcriptomics. Sometimes microbes are so similar that just looking at 150 base pair fragments doesn't really tell you a lot. But when you expand your view into fragments that are thousands of base pairs long, that's where you start seeing a difference. And because those longer reads start to discriminate and have, I'd say, more discerning power regarding which organism you're looking at. So we're using our long read technology to basically improve on Illumina sequencing in terms of read length. I should also mention that error rates in Illumina sequencing, and certainly in PacBio and Oxford nanopore sequencing, pose a major challenge in differentiating closely related species, and I'll show you how Loop Genomics is improving not only on the read length, but also on error rates compared to any other platform today. Loop's microbiome sequencing technology is also quantitative in the sense that, in contrast to Illumina sequencing, where people are today calculating the relative abundance of the species in their samples simply by counting the abundance of the reads off the sequencer, I'll show you today how that practice is fundamentally flawed because of a phenomena called PCR bias. What we're doing with loop sequencing is eliminating all of those PCR biases from microbiome quantification through single molecule counting technology. And I'll say a few words about that in a few slides. And then finally, the last aspect that I'll mention is that we have developed multiplex workflows that enable you to process anywhere between 24 and 96 microbiome samples in a single tube workflow that greatly reduces hands-on time in your sample prep workflow. Okay, so let's move on now to actual science and data. This slide here attempts to describe the fundamental technology underlying Loop's long-read sequencing technology. This technology, description rather, is a generic description that describes how we transform short-read sequencers into long-read sequencers for any DNA sequencing application. I will explain this through 16S sequencing, but you can generalize this description to how we do long-read sequencing at Loop Genomics for essentially any application, be it genome sequencing, shotgun metagenomics, full-length mRNA sequencing, or 16S sequencing. Okay, so starting at the top here, I'm assuming you can see my cursor here. Here on the left, you can see these are the Loop Genomics kits that we are now selling for 16S and 18S sequencing, for long-read sequencing. Everything that you need to bring you from extracted genomic DNA all the way to a library that's ready to load on an Illumina sequencer is included in this kit. So you don't need anything else besides your sample if you have this kit. It includes every reagent to make a sequencing ready library. Okay, so the first part of the Loop Genomics prep starts from a barcoding reaction. In this barcoding reaction, we assign a loop-specific adapter that's marked in this kind of bluish barcode icon. This loop-specific adapter includes many different DNA elements in it, but most importantly, it includes a unique molecular identifier, also known as a UMI. A UMI is a barcode written in DNA. So each loop adapter has a unique molecular identifier written in DNA, a barcode. And each DNA molecule in your sample, let's assume that this is a molecule from your sample, a 16S molecule, a full-length 16S molecule, say 1,500 base pairs long. Each molecule in your sample is attached to a loop adapter that has a unique UMI. So every molecule in the sample is tagged by a unique molecular identifier. The next part in our prep, which is really the heart of our chemistry and the reason that we've been able to transform short-read sequencers into long-read sequencers, is demonstrated in the next step. So we've developed an enzymatic operation that essentially, you can think of it as kind of a copy-paste operation in DNA, where we're able to take this barcode here and copying the barcode into multiple positions within the same molecule, but not to neighboring molecules. So it is a intramolecular barcode distribution step, where this barcode gets copied and pasted randomly into multiple positions within the same molecule, but not to neighboring molecules. So now that and obviously we're zooming into only one molecule here, but this happens in parallel over many, many millions of molecules in many different samples in multiplex in a single tube. Once these barcodes have been distributed intramolecularly, we've essentially now solved the major problem in long-read sequencing, because what we can now do is take these molecules and prepare them with for sequencing using regular short-read sequencing prep, right? So if we now break these molecules down into short reads, these are the short reads, we can, and sequence them on an Illumina sequencer, we can then take all the short reads that share the same barcode, right? We cluster all the short reads that share the same barcode into a single bin or a single cluster, and then use the sequence in those short reads in order to reconstruct the sequence of the original long DNA molecule that was tagged by that same barcode. So again, I'll just run you through this really quickly because this is a critical point to understand. Each long DNA molecule in your sample, say this is a 16S sample, or this can be a 10 kb fragment from a shotgun metagenomics sample. Each molecule is attached to a unique molecular identifier, a barcode written in DNA. Once the barcodes are attached, each barcode is copied and pasted into multiple positions within the same molecule, but not to neighboring molecules. And then these molecules, these long molecules with the barcodes inserted into them, are prepared into a short read library. The short read library is sequenced on an Illumina sequencer, and then through our software analysis that runs on Loop Genomics' cloud, we group all the short reads that share the same barcode into a separate bin, and then we use the sequences, all the short reads that share the same barcode, in order to reconstruct de novo, using de novo assembly pipelines, into the long molecule that was tagged by that barcode. I hope this part is that I've explained this sufficiently, and you can ask questions about this process later on in the chat section. Okay, I can see that we already have one question. How does this affect error rate when Illumina has issues with crosstalk already? That's Charlotte, first of all, thanks for that question. And I have a separate slide describing how our procedure affects error rate, but I can give you kind of a spoiler alert that it essentially reduces the Illumina error rate 40-fold compared to Illumina traditional sequencing. And I'll get to that in a few slides. Okay, I should also mention that the software analysis that takes all the short read data and transforms it into long reads, and the software for taking those long reads and doing the downstream microbiome-specific analysis on them, for example, classification, quantification, OTU analysis, is all part of our computational pipeline that we run on our cloud. And in that entire set of computational analysis is included in our kit. So anyone that purchases the kit gets a free one-time license to analyze the data from that kit on our cloud pipeline. All right, moving on now. Just to summarize what I'm going to show you so far. So I'll show you how our multiplex workflow reduces hands-on time about 10-fold, how our long-read sequencing that I just showed you how we're able to accomplish increases read lengths up to about 50-fold. We're getting reads that are up to 10,000 base pairs in shotgun metagenomics and genome sequencing. And obviously in 16S sequencing, we're getting full-length reads that are about 1,500 base pairs long. The fact that we're reading the entire length of the variable region, so we're reading all the V regions, V1 through V9, both in 16S and 18S sequencing, means that we can classify molecules down to the species and strain level, and I'll show you data about that. And that's in contrast to regular Illumina sequencing that only reads 1 or 2 variable regions. And The fact that we're using unique molecular identifiers means that we can quantify based on single molecule counting and eliminate PCR biases, and I'll show you a few slides about that. So these are the major topics that we're going to cover, and I think that these are the major topics that are really important for people doing 16S sequencing. Okay. And if there are other topics that I haven't addressed during the call and you find interesting, please mention them in the chat section, and I'll try to address them if we have data on them that is not included in this presentation. Okay, so the way to do microbiome sequencing with Loop is to basically get this kit that we're now selling on our website. As I mentioned, it includes everything you need to take you from extracted genomic DNA all the way to a library that's ready to load on a sequencer. Once you load it on the sequencer, you take the short reads off of the sequencer, you upload them to Loop's cloud, and we generate the data for you. So really, there's zero upfront cost in testing our technology. You don't need to get any additional hardware. You can use your existing Illumina infrastructure, so there's zero upfront cost. You leverage your existing infrastructure. You simply replace your existing sample prep with Loop sample prep, and our sample prep probably doesn't even cost more than your existing sample prep setup. So one final slide about kind of the high-level workflow. Our kit receives as input extracted genomic DNA from either 24 or 96 samples. That is used as input into our kit. You prepare your libraries using our kit. The entire process takes a single day with probably between 1 and 2 hours of hands-on time. Most of it is cycling time. You load your library on the sequencer, upload it to Loop's cloud, and within a few hours, you get a link to download your results. Your results will be in the format of FASTQ and FASTA files of the long reads. And additionally, a set of analyzes each one of your samples. For each one of the FASTQ files will also generate OTU tables, stats files with classification, quantification based on single molecule counting, and I'll show you some data about that. Okay, so let's start with the read length and its effect on microbiome classification in 16S sequencing. So what you're seeing here is a read length histogram of a Loop genomics sequenced sample. So this is a complex microbiome sample that we've sequenced once with Loop sequencing and then again with Illumina sequencing with both V3 sequencing and V4, V5 sequencing. So Illumina sequencing that covers 2 variable regions, V4, V5, and 1 variable regions with V3, and the same sample sequenced using V1 to V9 sequencing with Loop genomics kits. So what you're seeing here is the read length histogram of the reads from the 3 different methods. For Loop sequencing, you can see that molecules, so on the x-axis is read length, and on the y-axis is simply the number of reads, or the percent of the reads. And what you can see is that the Loop reads in red center around 1,500 base pairs, and that's because that is the natural length distribution of 16S molecules in nature. The reason that in Illumina data you see kind of a sharp peak is because that's simply determined by the read length of their instrument. Okay. Why should you care? Why is this important for microbiome sequencing? So you can view variable regions as like barcodes for bacteria. The more of them you read, the higher their phylogenetic resolution. So as you can see on the bar chart here, this is the bar that shows the Loop analysis, and these are the 2 bars that show the Illumina analysis. So what you can see is that a much larger fraction of the sample is quantified at either the species or strain level in Loop sequencing compared to Illumina sequencing. And the reason for this is because we're reading all 9 variable regions and basing our classification on 9 variable regions instead of just 1 or 2 variable regions. So that is the reason why Loop sequencing in 16S gives you greater phylogenetic resolution compared to Illumina sequencing. And obviously, I'm sure all of you know, there are specific strains and species that simply cannot be differentiated when you're just looking at one or two variable regions. There are species and strains that have identical V4, V5, or V3 regions, and it's only when you start looking at the entire set of 9 variable regions when you start seeing the difference between all the different species in the sample. Yeah, I'm seeing a few questions here. So Charlotte, what is the profile data graph based on? Human gut microbiome? Yeah, good question, Charlotte. So this is actually a complex soil sample. So this is a sample from one of our agricultural biotechnology customers, and this is a complex soil sample. So typically, soil samples are approximately an order of magnitude more complex than human microbiome samples, but I can definitely reassure you that a human microbiome sample would look, at least the read length histogram would look approximately the same. Okay, a few more questions here. Yeah, so how does the analysis technology deal with the short reads and chimera potential? So our computational analysis technology is based off of our long reads, not based off of our short reads. And the way that this relates to chimera formation is that we're actually able to use the barcodes and this kind of cloud of short reads that cover each long read to eliminate chimeras. Because if there is a chimeric molecule, what we essentially will be able to see is that some of the short reads that have this barcode that belong to one molecule, and then another part of the short reads that have the same barcode would belong to a different molecule with a different sequence. So that actually makes chimera detection easier for us. So we can more easily, I guess, eliminate chimeras from the data because we have this extra information. We have multiple short reads covering each long read, and we can use them in order to see whether there are chimeras in the system. But generally, I can tell you that the chimera rate is extremely low in our system, so sub 0.1% of the data are chimeric. Okay, I'll answer one more question, and then I'll move on. Let's see. Okay, so I should mention that in our loop adapters here, there are two hierarchies of barcoding. One is the sample index, and one is the molecular index. The sample index is identical to all of the molecules that originate from the same sample. So I should probably zoom in here into the structure of our kit, and as you can see, there is a 24, or I should say a 96 well plate here. So in each one of the wells in this 96 well plate, there are the reagents for barcoding a single sample. So in each one of the wells, there is an adapter that has a single sample index. But the UMI fraction of that adapter is highly diverse. So for each sample, the sample index is constant, and the molecular index is highly variable, so that there are no two molecules in the sample that have the same barcode, the same UMI. So again, the sample index is constant per sample, and the molecular index is extremely variable, and it is there are no two molecules within a sample that would get the same UMI. Okay, so this is read length and how we're improving phylogenetic resolution through reading all 9 variable regions on an Illumina instrument for each and every molecule. The next issue that I want to, or feature of our kit that I want to address is PCR bias. So For those of you that aren't familiar with the concept of PCR bias, so 16S sequencing technologies today are all based on, all involve PCR at some point of the process. And PCR is an amazing technology, but it is not perfect. There are certain drawbacks to PCR, and potentially one of the largest ones is PCR bias. PCR bias is the process in which different molecules in the sample amplify at different rates during the PCR process. So if you're performing a, say, 30-cycle PCR, some of the molecules in your sample will be amplified in each and every cycle. So they will be doubled 30 times, but some of the molecules in your sample will only be amplified 20 times out of the 30 cycles. So that means that there will be 2 to the power of 10 difference in the abundance of those two molecules, and that's about 1,000-fold. So what we're seeing is that PCR bias is a huge issue in 16S sequencing. For each and every one of our sequencing runs, we can detect the PCR bias. And the reason that we can do this is because we can count the barcodes that we see, and also count how many short reads are associated with each barcode. So a barcode that was, or when I say barcode, I mean essentially a single molecule tagged by a barcode, we can see whether a molecule was amplified 30 times during PCR or 20 times during PCR. So this graph here essentially summarizes this data. So on the x-axis, these are single molecules that were tagged by barcodes in our barcoding step. And then on the y-axis, we're plotting for each one of those single molecules how many times it was amplified during the PCR process. So what you can see is that there's a huge PCR bias. So for example, this molecule here, say molecule number 2000, we see it about 100 times in the data. Whereas this molecule here, molecule number 12,000, we see that molecule about 1,000 times in the data. So we know for a fact that since these two molecules have unique UMIs, we know that they were both single molecules at the beginning of the experiment. But at the end of the experiment, we see this molecule 10 times more abundant than this molecule. And if we look at this molecule here, this molecule would seem 100-fold more abundant than this molecule here. And the reason for that is PCR bias. This molecule was amplified much faster or more times during PCR compared to this molecule. So, and this is the process that's called PCR bias. And it leads to huge biases in how we quantify our microbiome. Because if you just took the reads off of the Illumina sequencer and assumed that the read abundance correlates to the actual abundance of species in your sample, you would think that this species here that corresponds to this molecule is 100-fold more abundant than this molecule here. But essentially, we know that they were both single molecules at the beginning of the experiment. They had equal concentrations. So these are the types of biases that you currently have in your Illumina short read data, and these are exactly the types of biases that we are correcting for, because we can look at the UMIs, at the barcodes, and completely disregard how many times we see them in the data. Each UMI will only be reported once in our FASTQ file. So that's a fundamental difference in the FASTQ file that we generate, the long-read FASTQ file, compared to the Illumina FASTQ file. The Illumina FASTQ file will be full of these duplicate sequences, and you can't really tell when you're looking at a short read from an Illumina sequencer, you can't really tell whether this is a copy of something that you've already seen in the data, or whether this is an actual new unique molecule. In contrast to that, in Loop's data, since we're essentially single molecule counting barcodes, we only report every molecule once in the data. So every molecule we report is unique, and you'll never see that molecule again in the data. And this is a bar chart that shows why this correction for PCR bias is so important in microbiome research. So let me explain what you're seeing here. So on the left, we're basically taking a complex microbiome sample and then binning the species according to their abundance. So this bin here on the left is the bin that contains the 6 most abundant species. And the bin here on the right is the bin that contains the 200 least abundant species. And what we're plotting on the y-axis is the PCR bias, or in other words, the error in quantification if you're not correcting for PCR biases using single molecule barcode counting. So what you can see is, so if there's no bias, this value should be 1. And if the bias is tenfold, then this value will be 10. So as you can see, for the most abundant species in your sample, the quantification is actually pretty good. It's very close to 1. But as you move from left to right, from the most abundant species to the least abundant species, you can see how this error starts to creep up to the point where in this bin that has the 200 least abundant species, their error rate is between 2-fold and 20-fold. So that's a huge error rate between 2 and 20-fold for 200 species. Now, that is about 1/3 of the species in the sample. So that's a huge error rate in quantification of relative abundance for a very large fraction of the species in your sample. And this is a general feature of microbiome samples. There are always many more species at low abundance than there are at high abundance. And when you think about it, that kind of mathematically has to be the case because, for example, at 50% abundance, there could only be 2 species, right? So there will always be very few species at high abundance, but at low abundance, there could be many, many species. So we've sequenced many different microbiome samples, and we see this as a general feature in microbiome samples. There are always many more species at low abundance than there are at high abundance, and the error in quantification is always much higher for those low abundant species. So somewhat tragically, a very large fraction of the species in your sample have a very high error rate in their quantification. And this is exactly the sort of errors that we're correcting for in our single molecule counting technology. Okay. I see a question from Jeffrey. Do we need to amplify V1, V9 region first? And how do we control that the same DNA can get the same UMI? Great. Both great questions, and I can tell you that both of these issues are taken care of with the kit. So to answer your question, do you need to amplify V1, V9 region first? The answer is no, you do not need to do that. Everything related to tagging of V1 and V9 molecules is included in the kit. So as I mentioned before, the input into your kit is extracted genomic DNA. So you extract total genomic DNA from your microbiome sample, and that is the input into our kit. You don't need to do anything else. And then how do you control that the same DNA gets the same UMI? Again, you don't need to worry about that. That is exactly the chemistry that we developed that I described here. Our intramolecular chemistry means that every DNA gets the exact same UMI distributed along its length. So since every DNA molecule is tagged by only one UMI, we then take that UMI and the same UMI and distribute it intramolecularly. The fact that it's intramolecularly, that means within the same molecule, means that the same DNA molecule always gets the same UMI placed throughout its length. So Jeffrey, I hope that I answered those questions. If not, you can write some additional ones in the chat section. Okay, so that wraps up how we are improving microbiome quantification using our UMIs. So it's the same UMIs that we're using to reconstruct V1 to V9. The same UMIs are used for correcting for PCR biases and essentially doing single molecule counting pre-amplification for microbiome quantification. Okay, moving on to error rates. So generally speaking, the error rate on Illumina sequencers is 1,600 errors for every million base pairs sequenced, or in other words, one error for every 600 base pairs. That's a number that you can find everywhere in the literature. That's a number that everyone agrees on. What we're seeing in our sequencing, in loop sequencing kits, is that we can reduce that error rate approximately 40-fold. So error rate is reduced from 1,600 errors per million base pairs to 40 errors per million base pairs. Now, one might ask, how are you able to reduce the error rate in sequencing if you're using, how can you improve on Illumina's error rate if you're using an Illumina sequencer? So the answer to that question is actually relatively simple. Let me walk you through this. So as I mentioned before, we are constructing each and every long read in our system through I should say, sort of a cloud of short reads that cover that long read. So each base pair in our long read is constructed through what we call consensus construction. And we also do what's called consensus error correction. So if there happens to be a random sequencing error inserted during the sample prep process, most typically during the PCR process, that error in the vast majority of cases will only be present in a small subset of the reads. So, for example, if there's an error in the short read in this particular position, there will typically be multiple other short reads that span the same region of the long read and cover that same base pair, and they will not have that error. So through the process of what we call consensus error correction, we eliminate almost all of the sample prep introduced errors. And that process alone of consensus error correction reduces the error rate 40-fold compared to Illumina sequencing. So our reads have an error rate that's 40-fold lower than Illumina's error rate, and Illumina's error rate is already an order of magnitude better than PacBio and Oxford Nanopores. So that is essentially how we're able to reduce the error rate in Illumina sequencing, in long read sequencing, despite the fact that we're using an Illumina sequencer. Now, you might ask, why is this so important for microbiome sequencing? The answer to that question, I hope, is exemplified in this plot here. So let me explain what you see here. So this plot describes the sequencing of a synthetic community. This is a synthetic community sold by Zymo Research, and it has 12 strains in it. So in general, I should mention that synthetic communities that have a predefined composition, both in terms of their 16S sequences and in terms of their relative abundance of each species in the mix, are potentially the best way to evaluate new microbiome sequencing technologies. So they constitute basically a ground truth against which we can evaluate new sequencing technology. So there are two, I'd say, major providers of synthetic communities out there on the market today. So it's also important that that each of us don't use their own synthetic communities, because it's only that when you have a community, a standard that all the community uses, that we can compare results from different methods. So the two best synthetic communities sold on the market today, one of them is sold by Zymo Research. It's called Zymbiomics. It has 12 strains in it. And the other synthetic community is sold by ATCC, and it is a 20-strain staggered mix of microbial strains. So this specific plot describes the sequencing of that Zymbiomics synthetic community sold by Zymo Research. So Zymo Research publishes all the information that's needed about the synthetic community. They have online, they've published the genomic sequences of those strains, together with the 16S sequences, the copy number of each 16S, and the relative abundance of each species in the sample. What we did in this analysis is we sequenced that synthetic community once with loop genomic sequencing that has an error rate of 1/25,000, And then again, the same synthetic community with short read Illumina sequencing that has an error rate of about 1 to 600. And what we did is we counted the number of unique species identified as a function of the number of molecules or reads that were included in the analysis. And we obviously know the correct answer. The correct answer is 12. There are only 12 species in this synthetic community. So what you can see is that with Illumina sequencing, essentially the more reads you throw at this analysis, the more species you get. And the reason for that is because with an error rate of 1 over 600, essentially every second or third read has a sequencing error in it. Once in every few reads, these sequencing errors actually hit a real organism that's present in the database, because it is often the case that even a single nucleotide change can switch the classification from one species or from one strain to a different strain. So these error rates, I guess, overinflate the number of the species count in your sample. In contrast with loop sequencing, because the error rate was reduced 40-fold to 1 in 25,000, you can see that we converge much faster to a number that's much closer to the correct number of 12. I think we converge to 13 in this case. So I'd say that it's been a general theme in microbiome research for many years now, that we seem to never be able to saturate what's called the collector's curve. So the more reads we throw at a sample, the more species we get, and it seems kind of like a bottomless pit. But we think that a large fraction of this phenomenon is due to sequencing errors being misidentified as new species. And now that we have this reduced error rate in 16S sequencing, we can see the true diversity of these samples. Okay. So that is how Loop Genomics' reduced error rate is contributing to a more accurate account of the species in your sample and not overinflating the species identification in your sample, and I guess reducing the false positive species identification rate in your microbiome sequencing. Okay, so potentially the last scientific issue that I'd like to address is that the fact that we're distributing this loop adapter throughout the length of the molecule means that we're not only distributing the unique molecular identifier, we're also distributing the sample identifier. What that means from a practical point of view is that after the short barcoding step, which only takes 20 minutes, you can pool all of your samples, whether 24 or 96 samples, into a single tube, because the sample index is also distributed along the length of the molecule. So that information is held within the short read. So you don't need to carry forward your prep for each sample separately. Post barcoding, you can pool all of your samples together, and the sample index distribution step will take care of the rest, and you can multiplex all your samples into a single tube. That greatly reduces the hands-on time in processing microbiome samples. That means that it doesn't really make a difference whether you're doing 24 samples or 48 samples or 96 samples. The processing time is more or less the same, and that's because very early on into the process, 20 minutes into the process, all the samples are multiplexed into a single tube workflow. Okay, so yeah, so I think I'll just kind of run through all the remaining parts of the scientific talk really quickly, because I think I've already hit the major features of our technology. This is just to show that our kits are able to produce highly accurate relative abundance data across orders of magnitude of relative abundance. So this is sequencing of the ATCC 20-strain staggered mix that I told you about previously. This strain has 4 orders of magnitude difference in relative abundance between the different strains. And what you can see here is that we're able to quantify. So on the x-axis is the expected abundance according to ATCC, and on the y-axis is our measured abundance as calculated by counting barcodes. And what you can see is that within a single sample, we can quantify samples that have a difference of 4 orders of magnitude of relative abundance. And that's something that is extremely hard to do if you're not accounting for PCR bias, because typically the samples here at the lower end will be highly biased in their abundance because you're sequencing so few molecules of these, and some of them will be biased. Some will amplify very slowly, some of them very quickly, and those are exactly the species, as I showed you here, that are at low abundance that have very high quantification error. So what we're seeing in this graph here is that we're essentially able to quantify them very accurately. Okay, another major issue in microbiome sequencing in general, and in 16S sequencing in particular, is GC bias. Typically, species with high GC content amplify slower in PCR. We've worked, we've invested a great deal of work over the past 2 years in order to eliminate GC-related biases in microbiome sequencing. So what you're seeing here is, again, this is the same ATCC 20-strain mix. One thing that they've done is, in the design of this mix is they've designed it to have a mix of species with varying degrees of GC content. So you can see the blue line here describes the GC content of these organisms. And you can see the scale on the right here. So this synthetic community has species with GC content ranging from 30% all the way to more than 70%. And what you can see is the expected relative abundance of each species, and right next to it, the actual measured relative abundance using loop sequencing. So what you can see here is that across the gradient of GC contents from different organisms, the expected versus measured abundance is in line. So it doesn't matter if we're sequencing an organism that has, for example, here, a 70% GC content, or if we're sequencing an organism that has a 30% GC content, our quantification is not affected. And the reason for that is, again, the fact that we can account for PCR bias, because these organisms here with low GC content would amplify faster in PCR, and these organisms with high GC content will amplify slower in PCR. So with just a regular 16S analysis, you'll have great bias. This would be overrepresented, and this would be underrepresented. But with our analysis, since we completely disregard how many times something was amplified during PCR, and we just account for the fact that we read its barcode once, we're able to account for that GC content related PCR bias. So that's how we're addressing GC content bias in microbiome sequencing, both for 16S analysis, but also for shotgun metagenomics. Moving on to just reproducibility. So when using our kits, you can expect to have a high degree of reproducibility. So if you analyze the same sample with 2 different kits, you're going to get the same result. So this is the theoretical relative abundance as published by the manufacturer. And then this is the data analyzing that same sample with 2 different loop kits at 2 different points in time. And you can see that you're going to get the exact same result. So our kits are reproducible from batch to batch. And then looking at that in a complex sample and not in a synthetic community, you can see that we've analyzed a complex sample in two different kits, two identical kits, analyzing the same complex sample. And then we're looking at specific, the abundance of specific geni. So you can see that this axis here is kit number 1, this axis here is kit number 2, and every dot on this plot is a single genus from those samples. So you can see that the sample, basically the geni are kind of arranged along this horizontal line, which means that the relative abundance quantification was almost identical in both kits. And this goes to show that in a complex sample, if you're going to analyze a complex sample twice with two different loop kits, you're going to get the same result twice. And this reproducibility is highly important in microbiome analysis. Okay. All right, so just a few words about workflow. So you can go online to the loopgenomics.com website and download our user manual and our quick guide. They have all the information about our workflow. This is kind of just a snapshot, kind of a screenshot of our manual. This is the quick guide. You can see that this is a protocol that can easily fit within a single day, and most of the time will be just idle, basically cycling time. The hands-on time is about 1.5 hours, whether you're doing 24, 48, or 96 samples. But however, if you don't want to complete the process within a single day, there are several safe stopping points, as indicated here in the quick guide, that you can basically put your sample back in the freezer and continue the next day. So it can be one day or a 2-day protocol, just basically based on your preference. So again, you can log on to our website, loopgenomics.com, download the full manual or the quick guide, and circle back to us if you have any questions. In terms of, so this is again just taking out of the manual. This is what the workflow looks like. There's the barcoding step, which takes 20 minutes. After that point, you pool all of your samples into a single tube, and these are the times these are the incubation times. So if you add these up, you see that this can easily fit within a day. Between these incubation times, there's several spry cleanups and several just pipetting pipetting operations. Generally speaking, it's about 1.5 hours hands-on time. Yeah, so those are the major topics that I wanted to cover during our call. I should say that the LoopSeq kit is compatible with any Illumina sequencer. However, we recommend using either NextSeq, NovaSeq, or HiSeq, simply because they have sufficient reads to support the number of reads that we need to reconstruct the long reads, and the cost per base is much lower. So traditionally, microbiome sequencing has been done on a MiSeq platform, and the reason for that is because of the importance of covering multiple variable regions for every molecule. So the MiSeq has the capability of covering 2 by 250 or 2 by 300, which enables capturing 2 instead of 1 variable region. But the downside to that is that the MiSeq is very expensive per base pair. It's probably the most expensive Illumina platform per base pair. What we're now enabling to do is we're enabling the microbiome domain to move away from MiSeq into cheaper per base pair platforms. And the reason we're able to do that is because we're now able to get long read sequencing from a 2 by 150 platform. So you can now move away from the expensive 2 by 300 MiSeq to the more than order of magnitude cheaper other platforms like NextSeq or NovaSeq or HiSeq, and get even longer reads. So you'll be able to get full-length reads that cover V1 to V9 with a 2 by 150 run, and also get cheaper per base pair costs. All of this information is present on our website and on our in our manual. What you'll also be able to find on our website, if you go to the Learn section, there are a bunch of technical notes describing different aspects of our technology, if you want to learn more about our technology. And there is also an example data set that can be downloaded of synthetic communities. If you're unable to access that data, please feel free to shoot us an email or a message through our website, through the chat section of our website, and we'll be able to send you any information that you need regarding the kits or just example data sets for you to review. In terms of the output that we will provide, the data output, as I mentioned before, that data would include FASTA and FASTQ versions of the long-read data, together with downstream processing, which is microbiome specific. So OTU tables, classification, quantification information. Maybe I can just show you this real quickly. So we would provide things like these quantifications, just showing the classification and quantification at different levels at the kingdom, phylum, class, order, genus, and species level. So for each one of these samples, we would provide these visualizations. We would also provide a stats file where, in this stats file, basically any every row in this stats file is a single molecule from your original experiment. Every molecule has a unique ID. And then for each of these molecules, we specify what the read length is. So you can see that for each molecule, we're reconstructing full-length molecules. You can see that they're 1,500 base pairs long. This is the classification, or I should say the reference ID for that molecule. The length of that molecule in the reference database. And then, for example, how well our long read aligns to that reference. In this case, there's 100% homology between what we've sequenced and the reference in the database. And then, obviously, of course, the full taxonomy for that molecule. So this level of information is provided on a molecule by molecule basis. I'm reminding you, this is single molecule sequencing. So for each and every molecule in your sample, you will be provided all this information. On top of that, we're taking that and doing that, taking that extra step and doing the visualization and the classification, and also providing OTU tables. So for each one of your samples, we would provide you with an OTU table. For example, this is sample 7 in this run. You can see all the classifications and the abundance of every OTU. So every row here is an OTU. This is the reference ID for that OTU. This is the abundance of that OTU in this particular sample. And also for that OTU, we also give you the taxonomy for that OTU. Okay, so this is just kind of a partial description of the data files that we would provide for each one of your samples. Okay, I can see that there's another question here from Kevin. Yeah, any performance and cost comparison with Oxford Nanopore? Yeah, so we're actually now in the process of publishing a comparative analysis together with an academic group, actually a few academic groups, comparing loop sequencing data to both PacBio, Illumina, and Oxford Nanopore data. So hopefully I'll be able to, that will be published very soon, so we'll be able to share the manuscript. In the meantime, if this is very important to you, I can please reach out to me personally through the website through the website, and I can kind of give you a sneak preview into some of the figures in that paper. But basically, I can tell you that in terms of performance, in terms of error rate and quantification, we're doing much better than Oxford. because, as I mentioned, our error rate is 40-fold better than Oxford's and better than Illumina's, sorry. And Illumina's is already an order of magnitude better than Oxford. So we're about 2 orders of magnitude better than Oxford in terms of our error rate. And other costs, I guess the other major cost performance metric would be the actual cost per molecule. And in that respect, we're also several-fold cheaper than Oxford Nanopore. So yeah, I guess in those 2 major cost and performance matrices, cost per molecule and error rate, we're doing much better than Oxford. But again, reach out to me personally, and I can share some figures to show that. Okay, just checking to see whether there are any other questions that I didn't answer. Okay, I think that we've addressed the vast majority of questions. All right, so I should mention that Loop Genomics is going to be represented in Japan by Tomy Digital. So in the future, you will be able to get access to our kits and our services through Tomy Digital in Japan. In the meantime, for our Australian customers, please contact us directly. Until we secure a distributor in Australia. Oh yeah, I see that we have another question here from Atsuko. How many species can I detect with this kit? Very good question. So Atsuko, the answer to that question is essentially how many species that you choose to detect. So our sample prep kit enables you to tune how many single molecules you want to sequence. So if you have a low complexity sample in which you only expect to have 100 or 1,000 different species, you can tune the number of single molecules that you sequence so that you don't over-sequence your sample. In contrast, if you have a sample that has very high complexity, for example, a soil sample or any other environmental sample, then you can tune the number of single molecules that you sequence accordingly. So you can sequence anywhere between thousands of molecules to millions of molecules per sample. And there is a place in the manual and the protocol that tells you how you can tune that. So if you read our manual, you'll be able to see that there are specific explanations that guide you into how to tune the number of molecules that you sequence, and that will determine the number of different species that you're able to detect. So the cost of our kit is exactly the same whether you sequence 1,000 molecules per sample or 1,000,000 molecules per sample. What would be different is the amount of Illumina sequencing reads that you would have to throw at your sample. So the Illumina cost would be different, but the loop cost would be identical, the cost of our kit. And you can go through our manual and quick guide to see how exactly in the protocol we adjust for the number of molecules that you want to sequence. And if that's not clear and you have any additional questions, please feel free to reach out to us, and we're happy to hop on a call or explain further over email. I should also mention that we're currently offering loop sequencing as a kit, but also as a service. So for those of you that are interested in outsourcing your sequencing needs, Loop is also set up as a full-blown microbiome service sequencing lab. So you can simply send us your samples, your extracted genomic DNA, and we will process them for you and sequence them for you and simply send you data back. So you can work in one of two modes. One is you get the kit, you do your own sample prep and sequencing, and we do the data analysis for you. And the second mode is you just send us samples and we do everything. You send us samples and we send you back data. So that's the full service mode. So feel free to reach out to us with questions regarding the cost of the kit or the service. The cost of the kit is currently, it's up there on our website. It's $999 for 24 samples. that adds up to about $40 per sample. And the cost of the service is really just dependent on the number of samples that you have and the depth that you want to sequence that. So please reach out to us for specific quotes regarding the service. Okay, I hope that I covered the major aspects that you were interested in. I just want to let you know that we have now released our 16S and 18S sequencing kit, and in a few weeks we'll be able to release our shotgun metagenomics kit and our genome sequencing kits, both based on the same long-read single molecule sequencing technologies. So please be on the lookout for that. The kits that are currently selling on the website are the 16S, 18S kits. By the way, these kits, there are two flavors to that kit. One does 16S only, and one does 16S plus 18S in multiplex in the same reaction. So the 16S kit would classify and quantify only bacteria, and the 16S plus 18S would map basically bacteria, archaea, and fungi in the same reaction. So those are the kits that are selling on the website, and look out for our shotgun and metagenomics and genome sequencing kits that will be out in a couple of months. In the meantime, if you do want to sequence your shotgun metagenomics and genome sequencing samples with us, we are providing those as a service until we have the kits live. I can see that we have one additional question. Let me try to address that. Which kit do you, so this is a question from Atsuko. Which kit do you recommend for DNA extraction? Yeah, that's a good question. We actually don't recommend any particular vendor for extraction kits. We've seen a great variety of kits being used successfully with our sample prep kit. So essentially, any commercially available kit, like the ones from Zymo Research or the ones from QIAGEN or any other well-known vendor that makes kits for microbiome extraction, has been shown to work with our technology. So as long as you have high quality, purified extracted genomic DNA, you should be, with a commercially available known manufacturer, you should be fine. I hope that addresses your question, Zuko. Okay, so if there aren't any additional questions, I'd like to thank all of you that have joined the webinar. I'd like to also thank TOMI Digital for enabling us to kind of broaden the scope of audiences that were able to join today, and hopefully you'll be able to get access to the Loop Genomics kits through TOMI Digital very soon. And please stay tuned for our upcoming webinars about shotgun metagenomics, genome sequencing, and transcriptome sequencing, all based on Loop's single molecule long-read sequencing technology. So I'd like to thank you again one last time here from the San Francisco Bay Area. Have a great rest of your day, and we'll talk to you soon. Bye-bye.